and thank you for inviting me here. Yeah, may I have the next one? Yeah. See, I was thinking about talking something about structural characterization and therapeutic aspects of the cyanobacterial phycobiliproteins. You all may be aware about that. Cyanobacteria are considered to be the oldest branching group of prokaryotes, and they exhibit oxygenic photosynthesis like that of higher plant. And it is believed that the present oxygenic environment is mainly contributed by the cyanobacteria. They are one of the major contributor of the for having the oxygen in the environment. And you can see, since I said that they are the oldest branching, so they evolved almost about 2.5 billion years before, or maybe earlier to that. And they are found almost everywhere. Whether you talk about, are you able to hear me? Is my voice is okay?
Yes, sir. Yes, sir.
Yeah, thank you. Okay. So what I want to say that they are found almost everywhere. They are present in the aquatic environment. They are found in the terrestrial. Not only that, they are also found in the what is called that. Yeah, something is going wrong. Is it okay? Okay. Okay. So they
Just
are
closed my sound, sir.
okay. Okay.
Like disturbed.
Okay. So they are also present under the extreme condition, whether you talk about the polar regions or a hot spring, or you can have the what is called that saline desert. They are found everywhere. And since they were evolved so much, they have experienced such a variety of stresses. And since they have evolved, or you can say that their genome has experienced such a variety of stresses, they have positively pressure, got evolved in such a way that they develop certain molecules which provide them to cope up such environment, which includes, of course, pigments, biofuel, many other things. There are many molecules which are synthesized by the cyanobacteria, and they provide some sort of a protections to them. So, may I have the next one? What basically you can have next one again. Yeah, yeah. What basically we do that we try to isolate the cyanobacteria from variety of environments, whether the marine environment or the freshwater environment. And since I'm a person involved with the environmental biotechnology, so obviously I work with the environment contaminated places also, so industrial pollution and other things. So we collected the samples from that, and we try to purify these cyanobacteria from such environment and get purified form, and they try to maintain in the laboratory. And these cyanobacteria basically we exploit for the at present. There are other applications also we have started work, and in fact one of my colleague Dr. Bhautosh Kikani is here. So we also try to use these cyanobacteria as a sort of not only for the pigment production but also biofuels, and we exploit the under the circular bioeconomy. One of the portion aspects which we are trying to do is that we try to go for the phycobiliprotein purifications. Once they purified in the extremely pure form, we go for structural characterization and try to see the therapeutic. In addition to these protein pigments, we also try to get the mycosporin like amino acids. These are specific molecules, and they to be, of course, we are not able to cultivate those mycosporin amino acid producing organism in the lab, but we directly collect it from the environment and try to purify the mycosporin. And they have a lot of applications. You will see later on in my lecture. So we try to exploit them for a variety of purposes. May I have the next slide, please? As I said that cyanobacteria basically carry out the oxygenic photosynthesis similar to the higher plants. That means that they also have a photosystem, one photosystem, two. They also show the photolysis of water. There is an oxygen evolution. But they differ in the what is called light absorbing capacity. In case of plants, they have a very narrow window, starting from about 670 to almost about the 720 nanometers. That particular wavelength they absorb, that is mainly because of the chlorophyll. Whereas when you talk about cyanobacteria, they have a wider window of absorbing photons or light radiations. Starting from almost about 450, you can say it can go up to almost about 750. Such a wide range there, and that is only because that they contain, in addition to the chlorophyll, we call them phycobiliproteins. These phycobiliproteins are present in that, and these proteins are responsible to have a wider range to capture the light energy. And that's all. So there they expand what is known as, yeah, yes, okay, please have next one. What are these phycobiliproteins? Phycobiliproteins basically are the colored fluorescent pigments, and they are made up of two polypeptides. Two dissimilar, I would use the word dissimilar polypeptides, brilliantly colored proteins. They are, we call them alpha and beta subunit. Each subunit is about 20 kilodalton, having about 160 to 180 amino acids, and they have a different amino acid sequence. Even though they have a different amino acid sequence, their folding patterns is quite similar to that. You can have a maybe somewhere in majority of the cases 7 alpha helix, and in addition to this protein, they also contains a chromophore group which is attached to this alpha and beta subunit through the sustainable residues. May I have the next one? Yeah. Based on the type of chromophore group they have, so they are characterized to three groups. We call them phycoerythrin, phycocyanin, allophycocyanin. So obviously, their what is called the light absorbing property varies. So we have a phycoerythrin which is a bright pink in color. We have phycocyanin which is dark cobalt blue, and aqua blue is a allophycocyanin. So you have a similar patterns. That's one. We have the next one. Yeah. Like any protein, obviously there is a gene corresponding to us. So we call it A gene and B gene. A stand for alpha, B stand for the beta. So you have a corresponding, and there could be multiple copies of the genes. So these genes obviously carry out the transcription followed by the translation, and they synthesize these proteins. What you get is apoprotein. But you have another molecule, what is known as heme, as a precursor or starting molecules, which through the various reductase process or enzymes, you can call it, from various different types of chromophore, and these chromophore group get attached. Depending upon the type of a chromophore group attached to the alpha and beta subunit of the phycobiliproteins, we can have a phycoerythrin, phycocyanin, allophycocyanin, and so on. This is what actually, and this basically once they form the chromophore get attached, then through the lyase enzyme, finally the complex is formed. May I have the next one? Basically, though we call it as it is made up of two subunits, alpha and beta. Alpha and beta subunit, though they are dissimilar in terms of the sequence concerned, though patterns may be the same, they join together, and this is known as heterodimer. And these heterodimers, again, three such heterodimers, again combined together, forms a trimer. In case of allophycocyanin, the trimers get arranged one above each other, and they form the bottom. They at the base, they will occupy the specific location. On the top of this base, you have again these trimers. They are also in case of phycoerythrin and phycoerythrin and phycocyanin. These trimers, they again back to back, they combine, forms a hexamer, and the hexamers are stacked together. So what you have that you have at the bottom, there is an allophycocyanin. From this bottom, you can say it radiates in a perpendicular manner. First phycocyanin followed by phycoerythrin, and forms a big complex, a giant complex, very big giant complex. And in addition to this, and already each of the subunit is already attached to the chromo. So you can call it a chromophore appended alpha and beta subunits. So it forms a big complex. And that big complex, which is giant molecules consisting of about 1,000 to 2,000 or maybe even several hundred thousands kilodalton unit, such a big thing, we call them phycobilisome. And this year only 2024 in the Nature, the paper appeared where they have shown the assembly of the whole phycobilisome. It is from the UK scientists who did good work on that. So that's how it forms a big complex. Now, this phycobiliprotein, yeah, next one, you can go ahead. it has multiple application. It has multiple application because it is fluorescent, dark in color, so it is used in the food industry, whether it is in the jelly, whether it is in the ice cream, or you can use it. And it has only amino acids, so there's no harm. It can also be found to have application in the pharmaceuticals, especially in the anti-inflammatory, hepatoprotective, even the what is called cholesterol reduction, and so on. It has. It also has application in cosmetic industry, specifically in the lipstick and other thing. You will come to know in my talk. It also has an application in the as a anti-aging, anti-Alzheimer. So it has a therapeutic application. Since these molecules are highly antioxidant, they are free radical scavenger, have application in nutraceuticals, and in some people have also used as a fluorescent probe. One of the major complications with the fluorescent probe which we face is that it is a much larger molecules. May I have the next one? Yeah. Yeah, please go ahead again. Again. Yeah. Please. Again, couple. Yeah. Yeah. That's it. So what we are trying to do that, we are trying to get the cyanobacteria from the environment. We go for the proper isolations. In fact, cyanobacteria algae maintenance is a tough task. Tough task in the sense that it is very difficult to maintain them in an axenic form. What I mean to say that it is extremely difficult to maintain these organisms in the pure form. They will always have bacteria as a contaminant. Even though you try to go through the plating, get the colony, then also they will have some bacteria associated with that. But anyway, we try to do that. Once we identify the cyanobacteria, these cyanobacteria are subjected for these pigment separations. Pigment. We go for the purification. Once we have the protein purified in the proper form, then corresponding genes are also taken out so that we get the sequence of nucleotide. At the same time, we also go for the mass spectroscopy, that is multi-top, so that you have a fragment analysis, so you know some of the amino acids also. At the same time, what we do, we go for the crystallization. Once the crystals are formed, they are subjected for the X-ray diffraction study. So you have X-ray diffract data, you have the fragment analysis through the MALDI-TAP, you have the nucleotide sequence available with you, and you have purified protein. It becomes comparatively easier to derive the three-dimensional structures. And by having the three-dimensional structures, you can go ahead with the docking study, see that which target it can get attached, and then you see some of the therapeutic application. That's what we are trying to do it. May I have the next one? Yeah. So for any protein, it's a conventional. There's nothing so great about the protein purifications protocols. These are all conventional. We either go for the ammonia sulfate precipitation or acetone precipitation, whatever it may be. You go for the precipitation. Here, before you go ahead with the precipitation, you require to do the lysis of the cells. And for cyanobacteria, the most conventional method of lysis of the cell is freezing thawing. You keep it at minus 70 degrees centigrade, bring it back to the 4 degrees centigrade, again go to the minus 70, bring it back to the 4 degrees. Like that, if you do the freezing thawing, perhaps it becomes easier, so that it will result into the crack formation. Since the pigments are present in the cell membrane in the actin, so it becomes easier to flush out the pigments. And once it comes out, you simply centrifuge it. What you have, the supernatant. Supernatant contains the pigment where the cell mass will retain at the bottom as a palette. And this supernatant is utilized for the conventional purification protocol where we use ammonium sulfate or acetone, whatever it may be. And then, so we follow the normal where we can go for the ion exchange, where we can go for the hydrophobic interaction, where we go for the molecular sieve, all those techniques we use, but we do some sort of little modification here and there. For example, one of the protocol which has become quite popular and people are using our protocol, that is use of Tritonics. This is surfactant. By having this surfactant in the beginning during the purification, we found that there's a resolution of this pigment. This three types of pigments get separated easily. You can do it. And then maybe you can have the hydrophobic interaction. And in case of allophycocyanin, we found that we use the ethodiene. It's one of the molecules which is used, and it helps in the resolution of that uh, allophycocyanin. So we are able to. So we developed several protocols, and they have been published also. That's what we did. May I have the next one? As I said that once the protein is purified, that protein is subjected. Yeah, of course, we get the information in terms of the gene. We isolate the genes. We get the nucleotide sequence done of that gene, alpha, beta, and then we go for the crystallization. And once the crystals are formed, initially, Dr. Gopal Jee, interesting to know is that initially we collaborated with, perhaps you may be knowing, Professor T.P. Singh. And this is for all of you information that
एम्स में है ना सर?
yeah, All India Institute of Physical Science. Yeah. Professor T.P. Singh is considered to be the one of the high-profile crystallographer. He is a man of physics turned biologist, and the number of molecules which are available in the protein database is the second largest in the world. The highest number, perhaps somebody is there in the Japan. Second largest is from the Professor T.P. Singh's lab. So we initially got involved with him, and he's a good friend of mine. Vijay. We interact quite often also, regularly. And then in his lab, we did the crystallization we did in our lab. Once the crystals were formed, these crystals were taken to his lab, and using his X-ray diffractions, we could get the diffractions data, and one of the first organism which we use in that's called the Formidium teni. And from this Formidium teni, we isolated phycoerythrin. It's only one pigment, one type of pigment out of the three. And this culture which was initially used was grown for more than 30 days. To our surprise, when this phycoerythrin was purified, checked, we noticed that this phycoerythrin is devoid of 31 amino acid from the N terminal. So it is a truncated protein. It's a small, having a molecular size of about 14 kilodelta. And that molecule was crystallized, and it was the first time truncated molecule, not only from our lab, from the India, which was crystallized and reported in the structural biology along with the Professor T.P. Singh. That was. So this molecule, initially we thought that it's not functional. It may not have ability to go for dimerization and complex formation. But we later on noticed that this 31 amino acid has nothing to do with the functional aspect. Rather, when the cyanobacteria are grown in a specific medium, and toward the end of after 25 days, 30 days, what will happen that all their nitrogen gets consumed. It's not a nitrogen fixing organism. So it gets consumed. So you try to utilize some other. So these 31 amino acids are utilized as a nitrogen source for their own growth. Advantage, it has advantage being a smaller molecules we can use as a probe easily. And that was our interest. May I have the next slide? Yeah. Very interesting that again this is the work which is done with an association with the Professor Gaurinath. You must be knowing he's from the JNU.
Yes, sir. Yes, sir.
He also happens to be a student of the Professor T.P. Singh. So we have another organism called leptoligmia. From this organism, we isolated a phycocyanin in the purified form. And that phycocyanin was crystallized. We went for the X-ray diffraction data, and we had many other information. So we could derive 3D structure of that molecule. And then we try to do the docking studies. Whereas all of you are well aware about that, there is an enzyme known as beta secretase. And beta secretase is one of the enzymes which involve in the amyloid proteolysis, which results in the plaque formation during the Alzheimer. This drug already available, which have a very high binding with that, and we notice that our molecules, these dimeric molecules, has a stronger affinity already with the known compound to the beta secretase in comparison to the known compound. So we thought that this will have a much better ability to do it. So we'll come to that. We try to exploit this system in the later. Then we had another organism. This was again the marine organism, which is a Formidio species. And from the same organism, we isolated all the three proteins: phycoerythrin, phycocyanin, as well as allophycosine. From the same organism, we could able to use for all the three proteins in purely form, and we could able to go for the crystal formation. And one of them is that phycoerythrin. And when we did the X-ray diffraction studies and based on other information, we could get the three-dimensional structure. And we noticed that one chain, alpha chain, which consists of about the 164 amino acid, and another has 184 amino acid, beta chain has one. And this alpha chain has two chromophore groups, which are attached to the cysteine 82 and cysteine 139. Whereas beta chain, consisting of about 184 amino acid, has three chromophore groups, of which one chromophore group is doubly attached at cysteine 48 as well as 59. Another one, cysteine 80, cysteine. In addition to that, we also found that there's an insertion of 9 residue amino acid between 146 to 152, and it has that uniqueness in the structure, which is different from than what already people have reported. May I have the next one? Similarly, we also did with the another molecule from the same organism, phycocyanin, which was also found to be hexameric structure. It showed the very unique property of having coevolution, having the phenylalanine, glutamine, and aspartic acid. And so what actually happened that suppose if the glutamine is not available at 33, you will find that phycocyanin will not have aspartic acid at 145, phenylalanine 20 will not. So these three amino acids, though they are different locations, they evolve together. And there is some sort of a functionality with reference to this. But this is also available. And this is the third. No, you can go ahead with the next one. Yeah. Which is the allophycocyanin, which was obtained from them. And a allophycocyanin showed the trimeric structure. And in this case, we very nicely notice that when you have a dimeric molecules, that is the heterodimers, where the chromophore group are found to be available with almost about 51 angstrom away from each other. When it forms a trimer, they come closer and the distance between the two chromophores is reduced to 20 angstrom. This helps when they are in the complex form, they have a, for the energy transfer becomes easier. So when the photosynthesis has to be carried out, energy has to be transferred. So they help it in that. So that's how we could manage, and then we got it published in the last one. We have the next one. Now, what is happening that any system, whether it's a cyanobacterial system, bacterial system, human system, or any other higher organism system, in a normal circumstances, this continues very well. Whenever there is a stress, and that stress could be anything. It could be the environmental factor, it could be your dieting factor, it could be anything else. And whenever there is a stress, it results into the formation of reactive oxygen species, what is known as ROS. And these ROS, obviously, They are strong oxidant, and they, since being a strong oxidant, it will disturb the cellular functionality. It will create so many complications. For example, in this, it carries out the protein oxidation, it causes the DNA damage, it causes the Fenton reactions, it causes the lipid peroxidase, and like that. There are so many. Though cell also has some level of antioxidant, which may be in the form of the what is called the superoxide dismutase or catalase or many other enzymes. They try to cut down the reactive oxygen level. But if the pressure of oxidant is higher, then obviously it can lead into the various complications. May I have the next one? We believe that whenever there is a reactive oxygen species formation, it can lead into the aging. And this aging oxidative state can get into the, whenever there is an oxidative state, there is an unfolding of the protein. Depending upon the type of damage which has been made in the protein, there could be different types of reactions. For example, if the damage is very high, the protein will undergo into the degradation, what is called that autophagy. So you will find that ubiquitin reactions, so they will undergo into the degradation, or they will undergo into the protein aggregations. If the damage is not very high, there is a possibility of that it again come back to the normal because of the help of certain chaperone and other things, some proteins. So there could be. So we are trying to correlate the reactive oxygen species with the agent. May I have the next one? Yeah. We are hypothesizing, or you can say that, proving that whenever there is a reactive oxygen species formation or oxidative state, it can lead into the aging. So that means that to reduce that antioxidant, what you require antioxidant, which will help in the anti-aging. And we noticed that these phycobiliproteins are very high, strong antioxidant. So therefore, phycobiliproteins can be used as an anti-aging factor. May I have the next one? Yeah. This is a simple, just what you call that assay which we do with the oxygen scavenging, reactive oxygen species scavenging reaction, a FRAB assay, another thing. These are in vitro antioxidant assay, and they are compared with the standard one, like ascorbic acid. And we found that our molecule is equally antioxidant as that of the standard one. May I have the next one? Yeah. Now, to do this experiment, how about anti-aging? We have used an organism that is known as that is C. elegans. C. elegans is a nematode. Cyanobacterial elegans. And it is a very small nematode, and this has a several advantages. It is comparatively small, easy to handle in the laboratory. It does not require any specific medium. It requires what is known as nematode growth medium, and it requires E. coli as a substrate, as a food to the growth. So we have specific E. coli cell, that is E. coli 050, that is used as a feed to the C. elegans, and it can be grown in the petri dish in the laboratory. You require the BOD incubator, which is maintained at 20 degrees centigrade or 16 degrees centigrade. That's all. And it can be easily absorbed. Since it is a nematode, it has a very active mobility. It shows the bending. It shows the contraction of pharyngeal. So you can easily observe it even with the simple, what you call dissection microscope. You don't require any specific conditions. And there are mutants available. Like whenever you go for the higher organism, people don't directly do the studies with the higher system directly with the human being. Even with the drug discovery, the experiments are initially done with the animals. Sometimes we may have to go to the primates, like the what you call the monkeys and other thing, and then it goes to the human, then clinical trials. So here also, you can do some experiment with the C. elegans, and moreover, you have mutants available. Like in the higher organism also, we have a mice model for specific, like diabetes mice model, Alzheimer mice model, like that. Similarly, you can also have a models for the C. elegans. And there is a specific institute that's called Therapeutic Research or Institute Genetics Research, which is at Minneapolis, USA. And from there, you can get the differentiate. Can you go back? Okay. Yeah. Thank you. Yeah. So what we try to do that, it has a very comparatively short life, maybe about 15 days or something like that, 15, 16 days or 15 days. What we did is that we grow them in the petri dish, and this petri dish was known as nematode growth medium, and that is preceded with E. coli. So this E. coli acts as a fodder or food for the nematode, so they utilize. Now, if this, along with the E. coli, if you provide phycoerythrin in the medium, so along with the E. coli, the phycoerythrin also will be taken up by the C. elegans. And with the result, what we notice that the lifespan of the C. elegans goes almost about 20, 30, 40% increase. That's what simply. And not only that, you can see the lifespan, their lifespan is increasing. You can also notice various other physiological marker. For example, one of the marker which is used, that is called pharyngeal pumping counting. You can observe their contraction relaxation. And with the help of stopwatch, you can measure what is the number. And you can observe on different days. For example, 0 days, both were more or the same, whether it is a PEF day or without PE. But as the time passes, you can say that the pharyngeal pumping counting goes down. But reduction is much higher when C. elegans is given without phycoerythrin. But in presence of phycoerythrin, it has a better. So you can observe it on the 5th day, 10th day, something like that. So there is a, that physiological characteristic also can be observed very easily. Not only that, you can say the locomotion assay. In fact, Dr. Gopal Jee, I also did some small work in the Dr. Siddiqui's lab when he was in TIFR, and I was in the TIFR for a year also, as a postdoc fellow. And there also, he was trying to understand the behavioral aspects of the Drosophila. And then we did some experiment with it. So similarly, here then the locomotions also, you can see their bending pattern. And their bending pattern can make out what is the level of locomotion. So you can observe that very easily. And you will find that having the phycoerythrin, their locomotion action is better than without P. This is what actually happening. Then may I have the next one? Yeah. Then we thought that, okay, this is observed when you talk about the wild type or the what is called the normal C. elegans.
Sir, और कितने slide हैं sir?
Huh? Yeah. Hello, this is okay. Is it okay?
Yes, yes sir.
Yeah. Yeah. Now, what we did, we tried to. Acha, you want me to rush up also?
Yes, yes sir. 1 घंटे का generally देख रहे patience नहीं है researchers में.
Acha, okay. I will try to rush up.
Yes sir.
I will try to rush up. Forget, huh? Okay, do. So what we did, we tried to give the oxidative stress. This oxidative stress can be given by heat treatment. Normally they are grown at 20 degrees Celsius. You increase the temperature to 35, you will find that they will die soon. But in presence of P, their lifespan is increased. Similarly, you can give oxidative stress of hydrogen peroxide. May I have the next one? Like that, you could help. May I have the next one? Yeah, please go ahead. Okay. Right, go back. Okay. One of the things is that what we thought that there's one pathway which is involved that is known as insulin or insulin-like growth factor signaling pathway. It has there's growth factor, or you can say control effector, growth control, that's called DAF16. DAF16 is synthesized or produced in the cytoplasm, but it enters into the nucleus and it is able to activate those genes which are responsible to improve the lifespan of the C. elegans under the stress situation. But then this is regulated. In the upstream, we have age1, and again to the next to that, there is another upstream that's called DAF. So when this age1 phosphorylate the DAF16, this DAF16 is unable to enter into the nucleus and it doesn't have the ability. So we thought that does it have any effect on that? May I have the next one? So what we did with the DAF16, we have put the what is not green fluorescence protein, and also we have, when there is a green fluorescence protein is attached, you can make out how the movement of DAF16 from the cytoplasm to the nucleus. You can observe it very in the under the microscope. And then we try to see that, and then we use the mutant strain which are devoid of DAF16 or age1 or that, and we notice that when it is devoid of the DAF16, in presence of P, the lifespan of the C. elegans goes up. So that means that DAF16 is not found to be linked with that. May I have the next one? Next one. Can you give Gopal Ji, please next. Something is going. It's not moving. Okay. So we thought that you can have one more. Do one more. So we thought there's an heat shock factor which is responsible for that. Heat shock factor is also
forward jana hai, sir.
It's not moving. Okay. Forward कर रहा है. Forward कर रहा है. It is not coming. Okay. So heat shock factors can be found to be. Yeah, I think not moving. It's not moving. Okay. Anyway, so then we notice that when there's a mutant, and what did we do? We use the RNAi. We have E. coli which can produce the RNAi, double-stranded RNA, and that using the RNAi of E. coli, that E. coli was fed to the C. elegans so that HSF factors get locked down. So it will not express. Under that situation, if you provide the P, it doesn't. So that means that heat shock factors and the P has some relationship. And then we try to see it with the other molecule like mouse tips. What happens that whenever there is a stress given, whether hydrogen peroxide or some other herbicide, you will find that ROH generation is very high. So in a control, you can see that there is a lot of green fluorescence with the help of this dye. And in presence of P, the ROH generation is reduced. So that's what it helps. May I have the next one? A very interesting thing which I'll just come to that. Is it? Can you go next to that?
Yes, like this.
Next. It's not moving. Okay. Anyway, don't worry about it. Then we try to, don't worry about it. It's okay. Then we try to do some experiment with the higher organism, human fibroblast, and other thing, and we notice a similar phenomena. That means that P has, no, don't go back. P has a very interesting thing which we have noticed with this is that, see, you know that, if you remember, there is called the A beta secretase. And that A beta gene is responsible for the beta secretase production. It is in the Alzheimer. So what we did, we tried to get this gene, and this gene is placed into the muscle cells of the C. elegans. And since it is placed in that, and it is placed in such a way that you have a promoter which is called temperature sensitive promoter. Because of the presence of temperature, as soon as you increase the temperature, the gene will be expressed, and under that expressed condition, the C. elegans will get paralyzed. And we found that when the P is fed to the same C. elegans, it helps. It improves the movement of the C. elegans. So it delays the action. So that is something which then we crystallized in fact, we got the beta secretaries from the human also. We tried to express in the E. coli cell. We did the, what you call the codon optimization, proper expression, though it went into the inclusion body, but we tried to go for the denaturation, did something, and we tried. And unfortunately, it was toward my fag end of my career, and there was a big fire in the lab, and we lost everything. But anyway, what I want to say that, see, this is again because the time is running short, so I will not talk about the other molecules. These are the microsporin like amino acid. They are also present in the cyanobacteria, and they can be used as a UV protector.
सर स्लाइड आपको नहीं दिख रहा है?
नहीं, slide दिख रहा है तो UV absorbing वाला दिखाई दे रहा है. But we need not. I think it is last some of the slides we have missed. But anyway, don't worry. You can go at the end, no problem. So, see, UV, as you all know that because of the environmental conditions, the ozone layer is depleting. With the results, the UV radiations are falling. And this UV causes several, whether you talk about UVA, UVB, UVC, and they causes various complications. And the cyanobacteria have natural capacity on their own. And there are different mechanisms by which they somehow protect themselves from the UV radiation. And one of the mechanism is that they are able to produce small molecules. We call them microsporin-like amino acids. And these microsporin-like amino acids have found to be useful as a UV protector. So we did some experiment where we have shown that the microsporin can be used as a sort of a UV protector in the cream cosmetic. And there are some experiments which has been done with that, and we have shown that that's what happened. But our lab is basically uses the cyanobacteria. It's one of the work, of course, not the, because I'm basically environmental microbiology person. So I have main work on the bioremediations is a major task. But in addition to that, we also, but these days we are trying to use the cyanobacteria in the circular economy, where we can exploit the cyanobacteria for the pigment production. At the same time, we wish to use them for the lipid production, convert those lipids by esterifications to the biodiesels, get some other molecules like EPS and other things, and see that in the process, the contaminants are also removed. Of course, this cyanobacteria alone would not do, but then we do it in combination with the bacteria as a consortium, and under this combination, it works out very well. I think that's what I think. Time is running short, so maybe I think I can take some questions from you if you have.